Okay, so I think we're ready to start the next session. If everyone can, everyone can take their seats, I think we're ready to begin. Hopefully we'll be joined by... Okay, very good. All right, well, Jeff. So it's my pleasure to introduce our next speaker. Jeff Duyn is a senior investigator in the advanced MRI section at the NINDS and leads a large intramural research program at the NIH. He led the 7T effort at the NIH for many years and now leads the new 11.7 Tesla human MRI scanner effort at the NIH. Very happy to have him here today. He'll be talking to us about brain anatomical contrast at 7 Tesla.
Good afternoon. I'm very happy to be here and I'm very grateful to have been invited. Thank you for that. I just wanted to show you some history and also some research. It's going to be a little bit of a mix. And I put in this slide just to show you how far we have come with field strength, but also with resolution. But I'm going to show you that not only resolution is much improved since, has not much improved since, but also the contrast. So this story at NIH with the 7T has been, we were the third 7T magnet, I think. There were, of course, other magnets, high field magnets available. So I think it was the Keith Thorne's magnet, the 9.4, and there was Ohio State, 8 Tesla. They were prior to us. But NIH got in around 1998, the talks started about getting also into high field. And that was primarily driven by Alan Koretsky. Alan Koretsky was still at Pittsburgh. And he was adamant about high field with his work, previous work. He had always been at the edge of high field. And so he wanted to continue that. And I was quite skeptical, but thanks for his pushing, we actually obtained he first convinced Story Landis to get into this, and Story talked to the NIH scientific director, and then they jointly went into this in around 1999. And eventually we got the magnet in 2002, and imaging started then in late 2004. So we had lots of problems initially. The good thing was with this system was that this is the initial research, the early research group in the early or the mid 1990s. So the good thing was that we initially had already 16 channels. So we had done some work at 1.5T and 3T, where we wanted to first have better SNR and also have multiple channels, because parallel imaging was just coming around the corner. And also there were some tight-fitting birdcage coils that showed a much higher SNR. There was one from Cecil Hayes' group, and also Doug Ballon, and I think he was at NYU at that time. He had a very close-fitting coil where he showed 50% improvement in the center of the brain. So we were hoping with surface arrays to not only improve just under the surface coils, but also get an improved SNR in the center of the brain over conventional birdcage coils. And that worked out well. So once that worked in around 2000, 2001, we demonstrated that. Then we teamed up with Biomedical, and that has been a very fruitful collaboration with. As a result, in the mid 2000s, we had a 32-channel 7T array, and our 7T had 16 channels. So there was 8 channels more than they originally offered. Of course, we had the problem that we could only connect 16 coils, so we were connecting either the back half or combinations, just depending on their area of interest. And what we found is that we substantially improved SNR not only in the center, but also in the periphery, or not only in the periphery, but also in the center. And combined with the field strength increases, we found a 25-fold improved SNR over 1.5T standard birdcage coil. And that then we used to improve the resolution. Now in MRI, resolution is very expensive. because you need an SNR improvement proportional to the volume of the voxel. So if you want to make a voxel twice smaller on edge, you need an 8 times improved SNR. So you lose that quickly, but nevertheless, you also improve the detail that you can see, especially when there are structural changes, contrast changes at the edge of what you can do. So if you then are able to resolve those structures, suddenly you see them, you get a lot more contrast differences. And that point was made by Peter Bantantini as well. So not only the resolution improved, but also with field strength, you have increased susceptibility contrast, so that the resonance frequencies shifts that cause susceptibility contrast, they scale linearly with field. And with these developments in high field, we also got developments in susceptibility weighted imaging. And this is also a bit of history. So turns out phase images were already available in 1986. And they could just see the major deoxyhemoglobin or hemosiderin accumulations with brain tumors. They could barely see that, and the areas had to be quite big. But since then, we even see in normal brain high resolution structure and details that we haven't seen before with standard imaging. So when we made phase images, we saw all kinds of structures that we also somehow saw in the magnet images, but with very different contrast, and some of them much clearer. Especially the contrast really intrigued us, because we were really curious why these areas had very different frequency shifts. So we knew that the basal ganglia would have a paramagnetic frequency shift because they are loaded with iron, but some of the white matter fibers, they also really stood out against the background, and we didn't really understand that. And what we initially saw, there were some orientation differences. So fibers that run perpendicular to the field, they stood out against the background. So that gave us a hint, but we let that alone for a little while. So what we could do with the phase images is get very nice detail in some cortical areas, but it was actually quite variable. So in some areas we got a very nice laminar contrast, with others not. And this had partly to do with these orientation differences. But if you could then compare the whole improvement from 3T to 70 with magnitude images, and then if you add then that phase contrast on top of it, you see that you can even get better resolution than just looking at magnitude data. My lab has spent a lot of time trying to understand the contrast that we see in susceptibility weighted imaging. And what becomes immediately apparent is that there is sensitivity to iron and myelin. So if you look at a myelin stain here and an iron stain, you recognize some of the structures that you see here, especially the white matter fibers. Basal ganglia, you see there's a lot of iron, here dark here. But it was still quite, the correspondence was still kind of complicated. There was not like a clear one-to-one thing like we have iron here and myelin there. So what we then did is we did our own staining. This is Masaki Fukunaga's work, where he compared myelin and iron stains with R2 star, which is another type of susceptibility contrast. That is just a transverse relaxation rate. And what you see is you get a stronger transverse relaxation in the line of Gennari. And but the line of Gennari also has high iron, but also high myelin. So that doesn't really resolve the issue whether it's one or the other that causes this contrast. By doing iron extraction, we then later found out that it's really the iron that causes the intracortical contrast. Then there is also some increased R2 star in the subcortical U fibers, and density of myelin is pretty much stable there, or flat, but the iron is stronger there. So that suggests that iron causes that contrast as well. On a finer scale, you can see that white matter has kind of a speckly appearance in both the iron stain and also in the GRE magnitude. This is susceptibility weighted imaging. And we have directly attributed this to iron, also based on the iron extraction data. But this is still not well understood. So this is all over the literature, the iron literature from the 80s, but they don't understand why there is this speckly appearance. So this still remains an unresolved issue. Then in white matter, I was talking about orientation effects there. What you see in the myelin and iron stain is that some white matter fiber bundles, they have high myelin and low iron, and they also appear with a high R2 star. And that was a little surprising because we initially thought R2 star that is just iron, but because there is no iron here, then it must be something else. And it turns out that it is actually the myelin, and it's not the myelin density, but the myelin orientation. And this one was then further demonstrated in collaboration with Afonso Silva's group, where he had marmosets that he could put in two orientations into the magnet, and then we could make images at two orientations. And the fiber structure in marmosets is quite well known, and we did the DTI image. So around the optic radiations, there is two orientations of fiber bundles. One is going through plane here, and one is like transverse. And you can see that those fiber bundles react differently to the orientation change. So one goes brighter and one goes darker. And that's not only true for the R2 star, but also for the frequency. So in white matter fiber bundles, there is a strong orientation dependence. A number of papers have come out on this, and it turns out that this orientation dependence relies on the compartmentalization of the water. So because there is a compartmentalization, and these compartments therefore lead to a biased sampling of the magnetic field, you get different frequency shifts in the different compartments. And depending on which one contributes more, you get a frequency shift in the image. Now, this is partly this is just because of compartmentalization by itself, but there is also an anisotropy of the myelin. So the susceptibility of myelin depends on its orientation in the magnetic field as well. So it's quite complicated. But in summary, both effects, compartmentalization and anisotropy, contribute to this frequency shift in the white matter. So we don't only have frequency shift, but we have different compartments with different frequencies, and that affects it to start decay curve. So both in the magnitude and in the frequency or the phase of the curve, you see an echo time dependence that is different for the two orientations. And this provides opportunities to analyze these curves and say something about the compartments that you are looking at. It also complicates the analysis, and I think the latter part is actually more worrisome because the opportunities, we haven't really been able to use these very well, but the problems are that you then cannot really calculate the susceptibility based on the phase data. So quantitative susceptibility mapping is confounded by this effect. We have worked with the clinical folks in NINDS on a couple of disease applications. I'm going to show you two. So fairly early on, Francesco Bagnaro was interested in using the 7T. And it was just at the time that we had these phase contrast images, magnitude contrast images developed. And what she found is similar to what Peter showed. There is quite a bit of heterogeneity in lesions, reflecting iron depositions within and around the lesion, but also this the oxyhemoglobin in veins that run through lesions. So they're using that now for better characterizing the type of MS that is occurring and helping adjust treatment protocols based on that. So this is an example of veins running through lesions, and the UCSF group also has shown it, as well as Peter Balnatini earlier. Another application is ALS, where we also saw strong contrast changes compared to normals, with a focus on the motor cortex. So ALS is a neurodegenerative disease with iron accumulation, and iron accumulates throughout the brain, but most severely in the motor cortex. And we see an increase in the R2 star consistent with a higher iron concentration. And if you compare that with histology, the location of that increased R2 star corresponds to where we see higher iron on the stain. Now, over the years, we had lots of problems, but one of the most difficult ones were motion and respiration, and the effects that they cause in the images, and how they affect the robustness of the imaging. So one thing that we found early on is that there is strong phase changes with respiration. So the respiration cycle affects the field in the brain, but it's quite predictable. So based on the chest position, you can predict how strong those phase changes are. And that is shown here. So the chest position in yellow, signal phase in white. And if you correct for that, then you can stabilize that. So we went ahead and used the real-time shimming, because it turns out these phase changes are not uniform over the brain. They have a pattern. So you have to use non-uniform field adjustments to correct for that. And we have done that with second-order shims. So if you do that, you can prospectively correct for the phase based on the chest valve data, and the white trace shows when you do that. This is purely based on second-order shimming during the acquisition. And that improves image quality. So if you don't do this, and this is an example with a person with a pretty heavy breathing, the heavier breathing, the more effects, and you see that there is a lot of ghosting, and also the resolution gets compromised. And that gets resolved when you use this real-time shimming. There have been other approaches as well. This is a commercialized approach that company Scope provides. I don't have any stocks in that company. But they use field sensors around the head, and they measure the field, and they correct for these nonlinear field changes across time, and they can also improve image quality. Now, then there is another problem with motion. So motion itself corrupts case-space encoding in MRI. You have to take care of that. So you can do that by motion sensors. But another effect of motion is that it affects the field in the brain, because if your head moves in the field of the body, of course, local field changes, but also the susceptibility of the head. If you rotate that, it also creates phase changes just from the head itself. So you have to adjust for that. And you can do that by monitoring those phase changes in real time. So we do that with a navigator. Jane Liu in my lab has done that. And then he uses that to retrospectively correct that. Well, this is kind of a cheap approach compared to the camera that I just showed you, the field camera that I showed you in the previous slide. But it takes time. So you have to build it in each sequence, and it takes several milliseconds. So in that sense, it's MRI expensive. Jane thinks he can speed that up to a few milliseconds. And we have started to apply this method to our MS studies, again with collaboration with the MS groups at NIH. And this is an example of that. So this is an MPRH, and then on the right you have no correction, only a B0 correction. And then if you then do a global B0 correction and motion correction, you get this result. And if you do the full correction based on nonlinear shim distribution, you can better resolve lesions and resolve some of the artifacts in image. So here is a small summary on what we have learned from 7T. So we can see brain structures and lesion detail that it's hard to see at 3T. It's not really, maybe not true that you cannot see them at 3T, but you have to really already know that these details are there and go back to the 3T, and then maybe you see them. And this is especially true for cortical MS lesions. Speed up maybe. There are microstructure and orientation effects in white matter that you need to take care of if you want to quantitatively extract susceptibility information. Both iron and myelin contribute to the contrast. And for a robust clinical application, we have to do a correction for field effects associated with respiration and motion. If I have a few more minutes, I want to show you where we are going with higher field. So currently, by where we are, is there is quite a few systems already that are stronger than 7 Tesla. Yeah, these are more or less to scale. So I expanded the images to get sort of to the scale of these magnets. So at NIH, we haven't had only system that's still waiting for helium. This 1170 in France has been at field. I think they put it back down and they're going to go back up in the summer. So I'm going to just show you a little bit of where our 1170 project is and where we came from. So we started in 2006. We started talking with Siemens and magnets, but it took us a little bit before we finally got the system. So we ordered it in 2008, and then in 2011 we got it. This is also partly thanks to Bruce Rosen that was very supportive during our PSC reviews. He's on the board of scientific counselors for our lab. So we were happy to get the money. This is again a collaborative project with NIMH and NINDS. So 4 PIs are involved, just to mention that quickly. So it's again, Bandatini and Shen are the main developers, and Alan Koretsky and myself on the NINDS side. So this was arranged together with swapping out the old 70 for a 70 shield, because we needed a base. So the old 70 had to go, but then we still wanted to keep a 70. And to place that, we needed a shielded one. So we got the first shielded 70 in 2010, and then this is the 11.7 system. Now, we had an unfortunate event just a few weeks after we got the magnet. So it made field, but then we did some acoustic noise testing for safety, and those quenched the system. And this is due to gradient magnet interaction. So if you pulse the gradients really fast, you create energy in the magnet, and that can warm up the windings or can cause mechanical motions in the winding. It's not clear what really caused it, but nevertheless, we quenched it, and the magnet got damaged, so it had to be shipped back to England. And it couldn't be repaired, and it took us, there were some legal issues, and it took us a little while to make arrangement. And then eventually ASG built a new system, and that just arrived back at NIH in the summer, in April. So we are now currently waiting for helium to energize that magnet. So it already made field in Italy, so we are hopeful that it will work. I hope not to quench it again. I want to acknowledge all the people that have worked on the susceptibility imaging. At the beginning, we have also done fMRI, but I didn't have time to show you that. Thank you very much.